Farhad.
Thanks. Thanks everybody for coming. I want to thank the organizer to give us the opportunity to present our work. Today I'm going to talk about the identification of causal gene for complex trait. So as you heard in a previous talk, genome-wide association studies, or in short GWAS, are a very useful technique that you collect individuals who have the disease called cases and individuals who don't have the disease called control. You genotype them and then you perform some statistical tests. And the output of this statistical test will be the significant statistics for each variant. And you can plot it in this Manhattan plot, which the x-axis is the variant location, and the y-axis is the significant score for each variant. Now, GWAS was very successful to find thousands of variants which are associated with different disease, but the question that we are interested to ask is that which of these variants are actually causal? I have to emphasize that the word causal here shouldn't be mixed with the word causality that is used in statistics and introduced by Judah Purr in computer science. What we mean by causal is the SNP that is responsible for seeing the peak of a significant. So here you have a peak, you want to know which variant is responsible for that. Because technically there are three variants in a region. Variants which are not associated with the disease, so they are not causal. The variant that is responsible for the peak, and the variant that is not responsible for the peak, but is in LD with another causal variant, and that's why you see that peak. So you may ask, why this problem is actually hard? Why don't you just pick a significant threshold, like a red line here, and consider anything above that as significant? So we did that, and I'm zooming into one of these regions which you have a very narrow peak, and you can see there are too many variants that pass your significant threshold. So which of these variants are actually important, or which one is actually causal? You may ask why this is happening. Most probably one of these variants here is causal, but all the other variants are in LD with this causal variance, so that's why they seem to be significant. So people have tried to define fine mapping method. So the goal of fine mapping is that we want to validate only a subset of variant which is considered to be significant from a GWAS result in order to find the true causal variant. So you can consider that the fine mapping is a post-process on GWAS result in order to narrow down your search space on the variant that later on you can go and validate and find the true causal variant. So here I'm showing you a very simple example of a loci which have 35 SNPs, and the y-axis is the significant level. Now, if I give this region and I tell you this is the LD pattern, this gray triangle, and each diamond in this triangle represents the pairwise correlation between two pairs of SNPs. And the darker the diamond, the higher the correlation. So if I ask which variant is causal, you can ask your intuition, and based on your intuition, most probably you will pick the most significant SNP to be causal. And in this case, your intuition worked fine, because the LD was very simple, and you can see the difference between the z-scores, or the significant level, is high, so most probably this was the true causal. And how we know it? Because we simulated this data, so we know the truth. However, life is not that simple, and we can have more complicated regions. So here is a very complicated region. As you can see, the LD looks more complicated. You can see a lot of darker diamond. And if you look at the significance score, you can see a lot of SNPs have very similar significance score, and a lot of them pass your significant threshold. So it's not very easy to use your intuition. But what our intuition says that using the LD and the significance score, we should be able to figure out which are the causal one. So more formally, people define fine mapping method as, given the marginal statistics, can we find which variant are causal? And this problem is hard for two main reasons. One is the linkage disequilibrium, which means that the variants are not independent. Because if the variants are independent, you can break the problem to two simpler problems and solve them each one independently, which makes it very easy. And if there are two SNPs in LD, if one of them is causal, you will see the effect on the other one. So the LD makes the problem very hard. And the other thing that people usually ignore is that in one loci, you can have more than one causal variance. So before, what biology people were using were two simple methods. One of them was the top k SNPs. So this method picks the most significant SNPs based on their significant level. Now, k, if it's 1, you pick only 1. If it's k is 10, you pick the 10 most significant SNP, and you consider them causal. The other method that recently they have used is conditional method, which I will explain in the following slide. So conditional method is an iterative step. In each iteration, you pick the most significant SNP as causal, and after you consider that SNP to be causal, you recompute the marginal statistics of all the variant, given that that SNP is causal. And you keep doing this process that all the SNP significant scores follows below a threshold. So here I'm showing that complicated region. I'm showing the true answer to you guys. The red triangle are the two causal SNPs. So this region had two causal SNPs. So what the conditional method does, it first picks the most significant SNP, which is shown by the red arrow. So you pick that SNP to be causal. You remove its effect from the genome and all the other variants. So that SNP drops, and you pick the next significant one. So you can see by the two-step of conditional, this method fails to pick those two real causal variants. So you can see that conditional method, in some cases, you will have suboptimal answers. So what we proposed in our previous method is called caviar, causal variant identification in associated region. This is based on a concept called row causal set. So row causal set is a set that can capture all the true causal variant with probability at each row. And in practice, you want to set this row to very high values, like 90 or 95%. So your goal is to find a set that can capture all the causal variant with probability 90 or 95%. But I have to emphasize, this doesn't mean that all the variant in this set are causal. What it means that if you go and validate every variant here, then with probability 90 or 95%, you can make sure that you found all the causal variant in that region. So a trivial answer to this problem is that you pick everything as causal. You pick all the loci in that region as a row causal set, and with probability 100%, if there is a causal variant, you have captured. And obviously, this is not what we want. So among all the row causal variant, the causal variant which has the smallest number of variant is the most informative one. And this has another benefit, which will have minimum number of validation as well. So here I'm showing you that simple region. We run caviar, and you can see caviar only pick one SNP, and this is the 95% causal set, which means that with probability 95%, if you validate this set, which only have one, you will capture the true causal variants. Now, let's go over the more complicated region. This is the more complicated region that they have two causal variants there, and caviar picks 6 variants. And the black arrow shows the set that is picked to capture the 95% causal set. So how does caviar work? Caviar works based on a concept called causal status. So for one SNP, you can have two possible causal status. Either that SNP has an effect or it doesn't have effect. So it's a binary value. Now, if you have a region with M SNP and all of them are in LD, which means you cannot break the correlation between them, you can have 2 to the M possible causal status. Now, for each of these causal status, you can compute a likelihood. I won't go to the detail of that. You can look at in the previous paper. But now we can define a causal set. So a causal set is a collection of SNPs. Now, if you have a causal set of size k, you can define 2 to the k possible causal status for that causal set. So how does caviar perform fine mapping? Given a region with M SNP and a causal set which has k SNP, you can approximate the posterior probability that this set can capture all the causal SNPs using this simple quantification, which you sum over the likelihood of all the causal status for that causal set, which there are 2 to the k possible term. And you divide it by the likelihood of all the possible causal status in that region, which is 2 to the m. Now, if this ratio is greater than rho, let's say 95%, then this causal set is a row causal set. And obviously, you want to try different k and find the minimum one. So this work was actually published in Genetics. Now, what we are interested now, we want to see if we can apply this to model organism like mouse. But the problem is that the model organism like mouse, the stretch of LD is much larger than human. So what this happens is that a lot of variant seems to be causal. So performing fine mapping on the variant level for model organism like mouse is not very informative. So what people are doing is that when you look at the loci, actually there are multiple genes that fall in that loci that can be significant. And all of these genes are in LD with each other. So people, when they do fine mapping in model organism, what they are interested to find which gene affects the trait instead of which variant. And they group the SNPs or variant based on which gene they are. And they consider a gene to be causal if one of the variant in that gene is causal. So here is a simple example I'm showing. Here there are too many variants that can be considered causal. So this is a toy example. And in order to find which variant is causal, you may have to put a lot of computational costs. But it's much easier to say that, for example, gene 2 is causal. It's a much simpler problem than saying which variant. And actually, this is more informative to the biologists to go and validate if this gene is causal or not. Another problem that when you're dealing with model organism is the concept of population structure. So the concept of population structure is that individuals are related genetically. So when you're doing GWAS, normally in human, they assume that there is no population structure. It means that individual that is collected in the GWAS study either are unrelated or they will try to make them unrelated. And when they are testing, they test this simple model which they assume that the phenotype, which is a vector of all the individual collected for that, in all the individual in that population, follows this simple linear model, which mu is the phenotypic mean, and xi is the minor allele frequency for the ith SNP, and beta I is the effect of that SNP, and with some normal noise. Now what they're testing if the effect of that SNP is significantly not zero. And in order to do that, people use linear regression, which is the formula star. But they make an implicit assumption. They make the assumption that the Y are IID, means that individuals are independent. But if you have population structure, you can't do that. So in order to solve that, people use linear mixed model. So they add this term U, which tries to capture the similarity between individuals. And U is a random variable that follows a normal distribution with some covariance, and that covariance depends on kinship matrix K, which models the similarity between individuals. Now, if you want to figure out what is the effect size, you solve linear mixed model, which is the formula is here. And if you look at it, it's very similar to linear regression, with only difference that this sigma inverse is inserted between the two components. Now, if you look at closely, actually, if you break the sigma inverse to a matrix that get multiplied by its transpose, you can actually see that the linear mixed model can be seen as a linear regression when you transform your genotype and the phenotype using this matrix of L. So the correlation between effect size can be computed to be the correlation between only the transformed genotype now. So what we did is that we did simulation to figure out if this quantity that we showed that is true in theory, if it really works in practice. So we computed the marginal statistics that it corrected in population structure, and we computed difference to the genotype. And you can see there is a huge difference. It's not close to zero. However, when we compare to the case that the correlation is computed in a transform space, actually the mean is zero, and actually the variance is very small. So that means that this formula that corrects for population structure, it actually works in practice. Now what we did is that we want to see if caviar gene performs better than other method. So we simulated data using the real genotype. There are 3 data sets, but I'm showing you only one. For more, you can go look at the paper. And we implanted 1 gene to be causal or 2 gene being causal, and we run all the methods. Now, the x-axis here is the number of genes we allow each method to pick, and the y-axis is the recall rate. So you can see in the case there is only 1 causal gene, all the method performs similarly. However, when you have 2 causal genes, caviar gene perform much better than other methods. And this is actually expected because all the existing methods assume there is only 1 causal gene or 1 causal variant in the region. However, this is a simulation. We want to see how our method performs on a real data. So we have GWAS result for HDL phenotype for 3 different mouse studies, F2 cross, HMDP, and outbred. And we know there exists a causal gene called APA2, which was validated by previous people. And this is the Manhattan plot of chromosome 1 that I borrow from Nature Review from Jonathan Flint and Eliza Resky. So we run caviar on this 3 data set, and we treat HMDP slightly different because HMDP was designed at UCLA, so people have a lot of prior knowledge. So we treat HMDP with one that we gave all the genes that are input to caviar, and one we use the prior information to filter out some of the genes that they know that it cannot be causal. So this is when we use all the HMDP, and this is when we use the prior knowledge. Now, what is interestingly, there are 5 genes that is picked by all the runs, and these 5 genes are named here, and one of them is APA2. And the other 4 are the genes that are very close to them, and our collaborators think that they are, if one of them should be causal, if another gene except APA2 should be causal, it should be one of them. So by this, actually, we can save huge amount of time for our collaborators. Instead of spending years to filter out these genes, we can do it in a couple of hours. So with this, I want to conclude my talk that fine mapping methods are becoming very popular, and we propose a new method that extends our previous method, caviar gene, which can work on a gene level instead of variant level, and it corrects for population structure. With that, I want to thank the people who were involved with this project, Gleb, Wayun, and Bogdan Pasinik. And I want to thank my supervisor, Eliza Resking, and the funding agency, especially the ISMB Travel Fellowship. And thank you for your time.
How much power did you gain from your transformation in terms of simplifying your data so that you could zoom in on the gene of interest?
The transformation, you mean the population transformation?
Yeah, exactly.
So the population transformation, actually, you will lose a little bit power. If you're not, a lot of variance seems to be significant because you have inflation. So actually, then you feel that some variants are actually causal, but actually they're wrong because you forgot to remove the population structure. So you would get wrong result. It's not even like people were doing mouse GWAS and they got a lot of significant peak, but most of them got removed because they didn't correct for population structure.
Do you have plans to apply this to human data sets next?
Well, we applied the caviar to the human because the LD is much smaller, and usually there the data is corrected for population structure. So not the caviar gene, but caviar, yes.
Good. Do we have more questions? Over here.
I really enjoyed that, but it's kind of depressing. We spent 5 years walking to a gene using chromosome walking. You jumped to it in a few days.
Thanks.
So terrific, thanks. My question is, can it be applied to plant genomes as well? Have you any plans to do that?
To what?
Plant
Genomes. Well, I think plant, again, is like model organism, they have long LD, maybe even longer. So I think it will be more benefit for our method, actually, for sure.
Absolutely, that's really interesting. Thanks a lot.
Thanks.
More questions for our speaker? So this is really impressive. It's been implemented.
Yes.
Is this going to be available?
Well, caviar is online, and very soon we will put the caviar gene online as well. Yes.
Excellent. Great. We'll be looking out for it. All right. Let's thank our speaker one last time. And our third speaker for this first half of this session is Alex Corn.